Chair, for the introduction. I also would like to thank the organizers for giving me this opportunity to present our work. It's a great pleasure to be here and to hear the many excellent talks. Compared with other groups, we actually haven't accumulated a lot of data for this project, and I'll show you what we have now. So in this project, we used a specific compound, the PHA 767491 compound, which is a dual inhibitor of Cdc7 and Cdk9, and on the hepatocarcinoma, together with the traditional chemotherapeutics 5-FU, and found them they work synergistically to suppress hepatocarcinoma, both in vitro and in vivo. Let me start by introducing the rationale behind our study. Hepatocarcinoma, as we know, is highly malignant. It progresses very quickly and is prone to develop chemoresistance to traditional chemotherapeutics such as 5-FU. 5-FU is an antimetabolite type of drug. It's a mimic of DNA and RNA and induces S-phase block. As one of the mechanisms of chemoresistance of hepatocarcinoma to 5-FU, activation of checkpoint kinase 1 plays a very important role. After the CHK1 is activated, it induces DNA repair and facilitates cell division. MCL1 is one upstream regulator of CHK1. It is a pro-survival type of protein, similar as the survivin protein. And the one upstream regulator of MCL1 is the cell division cycle kinase 7, Cdc7, which has been used as a target for the targeted therapy of various types of cancer. So, the PHA compound was reported by Italian group in 2007 in screening of Cdc7 inhibitors. And later it was found to inhibit Cdk9 as well. Cdk9 is a cyclin-dependent kinase and binds to cyclins to regulate transcription. So in this case, 5-FU induces CHK1 activation and induces chemoresistance, and PHA compound will combat with this resistance by inhibiting Cdc7 and subsequently inhibiting CHK1 activation. So in our experiments, we use two types of hepatocarcinoma cell lines, BEL7404 and HUH7. We use these drugs, we use the 5-FU at different doses and add the PHA compound at a fixed dose of 5 micromolar, and found this 5 micromolar PHA compound inhibits both hepatocarcinoma cell lines synergistically with 5-FU in a dose-dependent and time-dependent fashion. We also analyzed the activation of the CHK1 in this figure A. By adding FFU, CHK1 was phosphorylated at the serum site 3, 4, 5. And by adding the PHA compound, the phosphorylation of CHK1 was decreased. But the overall expression of CHK1 hasn't changed. And also, we didn't find a change in expression of Cdc7 in our case using the HCC cell lines. We also examined the expression of the upstream regulator of CHK1, which is MCL7, and found the level of MCL1 is decreased at both the protein level and the mRNA level. And we examined the apoptotic rate induced by 5-FU and by a combinational regimen of these two compounds. We calculated the number of condensed nuclei as the apoptotic cells and found apoptosis rate was increased by combination of the regimen. And we also analyzed the cell apoptosis rate by a flow cytometry analysis using an Xn4-MPI staining and found and confirmed the increase of apoptosis. And we also examined the level of cleaved caspase 3 and cleaved PARP. They were both also increased using the after the combinational treatment. And we established a xenograft of HU-H7 tumor model in nude mice and administered a combinational regimen to the tumor models. And from the tissues, we examined the activation of CHK1 and the in situ apoptosis by the tunnel analysis, and also confirmed the activation of CHK1 and increase of apoptosis by combinational use of PHA and 5-FU. So we reached the conclusion that combination of 5-FU with the PHA compound, which is still an inhibitor of Cdc7 and Cdk9, exhibited a synergistic anti-tumor effect on human HCC cells through inhibition of CHK1 and MCL1 and promotion of apoptosis. So we are continue on these experiments for further mechanistic studies for the combinational regimen, and the PHA compound as a prodrug hasn't been subjected to the systematic in vitro studies for its anti-tumor efficacy in a broader spectrum, and we also need to study the toxicology of this compound. So I reach the acknowledgement. I thank the financial supporting from the National Science Foundation of China and National Science and Technology Pillar Program. And the data I presented were produced by Drs. Wei Li and Lin Jie Li, and Drs. Hongjiang Shen and Shijiang Shang also provided technical support. And these experiments are were done in Zhejiang University School of Medicine, Children's Hospital. Thank you for your attention.
Okay, thank you, Dr. Chen, for your presentation. So do you have any questions?
You have shown the decrease in the apoptosis via MCL1 protein. MCL1. The inhibition of apoptosis via MCL1. Now MCL1 protein also has isoforms which can serve as pro-apoptotic or anti-apoptotic. So are you planning to look into this isoforms of MCL1?
Isoforms of MCL1. Oh, we haven't checked.
It is very interesting study.
I will ask you for the advice.
I have a general questions. So you have a very good results, combine two drugs, two inhibitors. I mean, do you have any in vivo test for mouse model?
Yeah, the last slide I presented in vivo xenograft model. We established HU-H7 tumor model in nude mice, and after the tumor was grown, we sacrificed the mice. And unfortunately, because we had to wait for the tumor to grow, so we started with varying tumor size. So for these particular experiments, we didn't see the significant difference in the tumor size after treatment. But with the tissues, we observed activation of CHK1 and increase of apoptosis by the tunnel analysis. So we are planning to do another session of animal studies.
Okay, thank you, Dr. Chen.
I got one question for you, Dr. Chen. And you mentioned about the drug, all right, PHA is a dual inhibitor of Cdc7, right, and Cdk9. So what is the binding site of this inhibitor to the two enzymes?
I think it's still unknown because it's a relatively new drug. And this drug has been only studied in multiple myeloma and our study.
Is the structure of the PHA known?
The structure is known.
Can you do any docking experiment to see whether it's bind to the ATP binding site or not?
It can
Using
be done.
computer animations, right?
Yes, I think it can be done. We
In
have
many
done
drugs,
that.
right, if you know the structure and you know the target, right, you can do docking experiment to
Yeah.
probe the direct binding site, right?
Yes, exactly.
Okay.
But this compound has multiple binding sites.
Multiple binding sites.
Yeah.
Okay.
It has a broader anti-tumor spectrum
Mm.
compared with specific Cdc7 inhibitors.
Okay, thank you.